the great pleasure to welcome and greet all of participants of the 20th Brazilian Symposium on Medicinal Chemistry. I would like to invite the chairs of 20 BrasMedChem, Professor Claudio Viegas Jr. and Professor Ângelo de Fátima, and also the Director of Medicinal Chemistry Division of Brazilian Chemical Society, Professor Gustavo Trocini, to compose this opening table. First of all, as President of the Scientific Committee, I would like to thank our guests from Brazil and abroad who kindly accepted our invitation, and I'm sure we will have a great event with a high-level program, which I hope you enjoy, as I also hope that in a post-pandemic world, we can use scientific activities like this symposium to bring people together, allow colleagues to meet in person, and, if possible, resulting in fruitful new collaborations. Now, I pass the floor to the chair of the event, Professor Claudio Viegas Jr.
Ladies and gentlemen, speakers, Congress attendees, and all herein present, good evening. We would like to thank you all for your presence, in particular to the Brazilian and foreign guest speakers at the 10th edition of Brazilian Symposium on Medicinal Chemistry, BrasMedChem. It's a great pleasure to have you here as the main players in this Congress, which was carefully conceived to offer you a welcome environment to discuss and share the state of art of medicinal chemistry research. The city of Poços de Caldas, in the state of Minas Gerais, was chosen due to its natural beauty, touristic structure, and welcome atmosphere to celebrate the 21 years of the creation of this scientific event. Indeed, the first edition of the BrasMedChem took place in Minas Gerais state in 2001, and since then has consolidated as the largest medicinal chemistry congress in Latin America. We are thankful to honorable sponsors and the organizing committee and the scientific committee who made this event possible, especially during such a challenging period for science and society as a consequence of the coronavirus pandemic. On behalf of the presidency composed by myself and my friend Professor Ângelo de Fátima, I declare open the 10th BrasMedChem. We wish everyone the all best in these 3 days we will spend together.
In the seconds, I give the floor to the other chair, Professor Ângelo de Fátima.
Okay, thank you. The day has come. Is that amazing to get together again and to have here in this BrasMedChem our beloved meeting. And we are pleased to welcome all of you to this Congress, to this symposium. We have prepared a wonderful scientific program, so we are going to have in these 3 days of event the opening keynote by Dr. Sophia, a short course on drug design, 10 thematic sections on several areas of research in MedChem, a section dedicated to human in MedChem, ceremonies to grant BrasMedChem awards, BrasMedChem Bright Olympics, best posters, And also, we have scientific activities for the locals, such as a theatrical performance, sorry, but only in Portuguese in this case. And so, we hope you have a delightful time in Poços de Caldas. Thank you for your attention, and I return the floor to Professor Marcelo. Thank you.
Finally, I give the floor to the Director of Medicinal Chemistry Division of Brazilian Chemical Society, Professor Gustavo Trocini.
Distinguished speakers, colleagues, ladies and gentlemen, good evening. I would like to thank you all present for attending the 10th Brazilian Symposium on Medicinal Chemistry, BRASMEDCHEM, on behalf of Medicinal Chemistry Division of the Brazilian Chemical Society. I'm glad to be here to welcome everyone. After a long time without present events, we are here to exchange expertise, ideas, make networks, and establish collaborations, and of course, make friends and discuss science. The Division of Medicinal Chemistry would like to present a special thanks to our colleagues, Professor Claudio Viegas Jr. and Professor Angelo Di Fatima, the chair and vice chair of these events, for the accept the challenge and the organize the BRASMEDCHEM 2022. Thus, in the name of our society, I wish you all an excellent and profitable event. Thank you.
So, to formally begin the activities of the 20 BRASMEDCHEM, please join me to thank the participants of this opening table, as it is undone, and we move on to the next step of this opening ceremony, where I will have the great pleasure to introduce the speaker of our opening conference, Dr. Michael Sophia. Michael Sophia received his BA in chemistry with honors from Cornell University in 1980. He went on to receive his PhD in organic chemistry at the University of Illinois, Urbana-Champaign in 1984, working with Professor John Katzenellenbogen, working with the discovery of serine protease inhibitors. Next, he joined the laboratory of Professor Gilbert Stork at Columbia University as an NIH postdoctoral fellow and worked on radical cyclization chemistry on vitamin D total synthesis. Dr. Sophia began his industrial career in 1986 at ER Squibb & Sons, where he developed inhibitors of squalene synthetase and HMG-CoA reductase for the treatment of atherosclerosis. His work led to identification of the first potent substrate-based inhibitor of squalene synthetase. In 1989, Dr. Sophia joined Eli Lilly and Company as a research scientist. He spearheaded work in the area of leukotriene B4 receptor antagonists for the treatment of inflammation associated with asthma and delivered the clinical candidate LWA 293-111, which progressed to phase 2 clinical trials. In 1993, Dr. Sophia took his first leap into biotech when he joined Transcell Technologies as the founding director of chemistry. He eventually was promoted to vice president of research. At Transcell, Dr. Sophia pioneered the development of carbohydrate-based technologies for combinatorial chemistry and anti-infective drug discovery. Subsequently, In 1999, Dr. Sofia joined Bristol Myers Squibb, where he became group director of New Leads Chemistry. At BMS, Dr. Sofia built a lead discovery chemistry organization and global research capability that supported all research programs, and that led to a dramatic improvement in the identification of new chemical matter to initiate lead optimization programs. Dr. Sofia left BMS in 2005 to join Pharmasset Incorporation as Vice President of Chemistry and was later promoted to Senior Vice President of Chemistry. There he built the company's capability and programs around novel antiviral agents. His work led to multiple clinical candidates, including a number of agents targeting the hepatitis C virus. His work on HCV resulted in the development of the HCV NS5B polymerase inhibitor, sofosbuvir, that was utilized as a novel liver target product strategy. In 2013, sofosbuvir became the first interferon-free HCV cure therapy approved for clinical use. It quickly became the most successful drug launched in history and was designated by the World Health Organization as an essential medicine. With the acquisition of Pharmasset by Gilead Sciences in 2012, Dr. Sofia remained with Gilead for a period of time as Senior Vice President of Chemistry, but soon founded Encore Biopharma to search the cure for hepatitis B. Encore Biopharma became Arbutus Biopharma Incorporation, where Dr. Sofia is currently the Chief Scientific Officer and leads the discovery research and preclinical development efforts in search for an ABV cure. Dr. Sofia has authored over 100 publications, 12 book chapters, and numerous abstracts, and he is an inventor on more than 53 US patents and numerous patents applications. He is on the advisory boards of Chemical Engineering News, ACS Medicinal Chemistry Letters, and ChemMedChem. For his work in drug discovery, Dr. Sofia is the recipient of numerous awards. These include the 2014 Pennsylvania Bioscientific Achievement Award, the 2015 Heroes of Chemistry Award of the American Chemical Society, Foreign Policy Magazine's 2014 Global Thinkers Awards, The Economist Magazine's 2015 Innovation Awards in Bioscience, and in 2016, the IUPAC Richter Prize in Medicinal Chemistry, among others. Dr. Sofia, thanks to accept our invitation. The floor is yours.
Well, thank you for that wonderful introduction. And I want to thank the organizing committee, Claudio and Angelo, for the invitation, for the gracious, welcome here in Brazil. It's actually my first trip to South America. So my first trip to Brazil. And it was wonderful, and I thank them for the incredible hospitality. So today I'm going to talk about viruses. And I guess it's apropos since we've been spending two and a half years talking about viruses. And So I'm going to talk a little bit about viruses first, give you a background. So viruses are the smallest of organisms. They are alpha-kit intracellular pathogens. They actually do not make their own energy. They do not make their own proteins by themselves. They require the use of host enzymes to actually produce the viral proteins. The genomes are either RNA or DNA. They're usually encapsulated. And the components are assembled and do not replicate by division. So it's not like a normal cell, the way viruses work. So if you look at viruses, there are a number of viruses that cause chronic diseases or persistent infection. Usually this is a result of viral host immune control. So many of these viruses have very unique ways of controlling the host immune system so that they either are latent, meaning that they hide some way in a very low replicated state in the body, or they find ways to actually control the host immune response to initiate a state known as immune exhaustion. So some of the key viruses that actually produce chronic diseases are hepatitis viruses. They're hepatitis C, hepatitis B, and hepatitis D, or Delta virus. Now, if you look at hepatitis viruses, so they attack the liver selectively, right? They cause inflammation of the liver. They're the major cause, 50% of them are the major cause of all viral hepatitis in viral origin. And if you look at, there are 5 forms of hepatitis. There's hepatitis A, B, C, D, and E. Hepatitis B, C, and D are the only chronic hepatitises. The others, hepatitis A and E, caused by ingesting contaminated food supplies, are usually self-limiting in and of themselves and resolved by the body. But hepatitis B, C, and D are not generally resolved. They can run from an acute phase all the way to chronic phase of infection. So we're going to talk about hepatitis C and hepatitis B today. If you look at hepatitis B and hepatitis C, 500 million people are chronically infected with hepatitis B and hepatitis C globally. Multiple billions of people actually have been infected. Some of them resolve the infection in the acute phase. 1.3 million people die. Globally, 57% of cirrhosis and 78% of primary liver cancers are due to these 2 viruses. 80 to 90% of liver transplants are associated with hepatitis B and hepatitis C. And the majority of those chronically infected are undiagnosed and often asymptomatic for many, many years before the liver conditions begin to manifest themselves. In addition, if you look at all the other viral infections or infectious diseases that are occurring, you see an improvement based on treatment regimens associated with those diseases or public health improvements associated with those infections. However, unlike for example, tuberculosis, HIV, and malaria, what you see is a continued increase in the incidence of chronic viral hepatitis globally. In the United States, one of the problems is that people who inject drugs ultimately contract viral hepatitis through the use of dirty needles in common use. So I'm going to tell you a little bit about the story of hepatitis C. This is not going to be the focus of the talk, but this is sort of just to highlight what medicinal chemistry can do in a field and ultimately led to a cure today. So if you look at hepatitis C is an RNA virus. Okay? It's a plus-sense RNA virus, about 10 kilobase genome. It's got 6 different genotypes, so 6 different varieties of the virus itself. It's an envelope virus. There is no viral reservoir, meaning it doesn't deposit its genetic information anywhere in the cell itself, and it doesn't integrate into the host genome. The virus life cycle resides only in the cytoplasm, so it enters the cell and replicates its genome, ultimately producing new virus, and no entry into the nucleus of the cell. Now, one of the problems with this virus, it has a very poor proofreading function in the replication of its genetic information. And as a result of that, you have many variants, mutations that result from the presence of this virus. So, in around 2003, 2004, there was a big initiative to try to identify a non-interferon cure for hepatitis C. So the regimen for treating hepatitis at the time, hepatitis C, was the use of interferon therapy. Now, interferon therapy was a pretty drastic form of therapy to take. Interferon therapy caused neurological side effects. It caused anemia, severe anemia, flu-like symptoms for the entire 48-week course of therapy. In fact, because of the significance of the side effects, most of the patients never actually completed therapy and actually never achieved a cure. Now, also the problem is that only about 30 to 40% of any patients who actually went through therapy ever achieved cure in the end. So it was a very difficult therapy with not a very high percentage cure in the end. So at the time, there were a number of criteria that when we began to look at this problem, set out for ourselves. So the questions were really, what is the target that we're going to look at? And I'll show you there are a number of targets in the viral genome that would be interesting to target. How potent would this molecule need to be from a safety standpoint? Also safety, what is acceptable? Interferon was very bad. You know, how much better do you need to be from interferon for the medical community to accept it? Resistance, how much and how fast? I mentioned that you have a very poor proofreading function associated with this virus, and therefore a lot of resistance would arise on drug treatment. But how quickly was that going to rise? There's 6 viral genotypes, all of them slightly different. You really wanted a pan-genotypic agent. Interferon worked very well in genotype 1, but much less well on all the other genotypes. Combination therapy was viewed at the time to be the solution to most viral diseases, treatments for viral diseases. And that stems from successes in HIV, where you see multiple drug combinations actually were able to control the disease pretty effectively in a wide patient population. So what combinations were going to be effective here for hepatitis C? Could you eliminate interferon? Now, at the time, interferon was viewed as an immune stimulatory agent. Therefore, it boosted the host immune response to be able to attack the virus. Could you actually eliminate that immune stimulatory component and also eliminate a number of the side effects associated with that? And finally, because this was a liver disease, the virus only attacked the liver itself. Could you actually identify an agent that would target the liver selectively, therefore improving your chances of reducing side effects associated with that? So the medical community at the time believed that the ideal HCV profile was high efficacy, so greater than 90% SVR. So SVR is sustained virologic response after 12 weeks off therapy. Meaning no virus detectable in the blood at 12 weeks after this therapy was stopped. You need to be well tolerated, so low risk of any side effects. Convenient, so short duration of therapy, simple dosing, and then effective in a broad patient population, since this patient population was global and also very varied in the nature of the individuals who had the disease. So at the time, many of the leading thought leaders in the space did not believe that this could be achieved before the mid-2020s. So even not today would there have been a cure based on the speculation of thought leaders at the time. Now, when you look at HCV as a virus, it has 10 viral proteins. 7 of them were known as non-structural proteins, or the druggable targets. And there were 3 major druggable targets that were actually pursued in the whole spectrum of HCV drug discovery at the time. There were the NS3-4A protease inhibitor, the NS5A replication complex inhibitor. So this is not even an enzyme. It's a protein that is important in forming what is known as the replication complex of multiple proteins that come together in the endoplasmic reticulum to initiate replication of the viral genetic material. And then there's the NS5B viral polymerase, or RNA-dependent viral polymerase. So these were the 3 targets that people were looking at. Now at the time, when you looked at the protease inhibitor, there was a tremendous amount of work going on at the time, and this is in the mid-2000s. And they were very potent compounds, but the problem with the protease inhibitors is that they have very low barrier to resistance. So resistant virus would emerge really quite quickly. The other thing was that they were only shown to be effective against genotype 1 virus, so no efficacy against genotype 2 through 6, so that was a significant problem. When you look at the replication complex inhibitor, extremely potent compounds were being developed in this space, led primarily by the work at Bristol Myers Squibb, and then other companies began to parlay off of their work. But was interesting about this, although very potent, it had a very low barrier to resistance. So in fact, about 4 hours after the drug was given, you began to see emergence of significant resistance in patients. So when you look at the NS5B RNA-dependent RNA polymerase, we were very interested in this target for a couple of reasons. Number one, the nucleoside binding site was highly conserved across all viral genotypes, gave the possibility for producing a pan-genotypic